All right, so you should be let's see, open that. All right, so to make my mRNA sequence, I use my base pairing rules. So what binds to T? Who remembers? What binds to T? No one wants. Good job, Stephen. A. What binds to A? Jacob? What binds to A? It would bind to T, except, what is this? What am I making? I'm making mRNA. So I have to change that T to U. Okay. What binds to C? What binds to G? C. What binds to A? U. What binds to G? C. What binds to C? G. What binds to T? A. A. What binds to A? U. U. And then C. Okay. So this is the mRNA sequence that would occur by the transcription process inside the nucleus using the sense strand of DNA. Now, that's not the entire part of the problem, because now I have to translate that into a protein. So remember, my mRNA has codons that are 3 nucleotide sequences that correspond to an amino acid. Okay. So I suggest that you separate them just because it makes it easier. And then you use the chart by starting in the middle. AUG, AUG would give me methionine. So my first one would be methionine. My second one is CUC. Again, I start from the middle. CUC, and that's leucine. GAA, so I have GAA, that's glutamic acid. And my final one is UUC. So I start in the middle. U is my first base, U is my second base, C is my third base. So that's phenylalanine. So those are the 2 steps. The first one is transcription and the second one is translation. All right, so now that we've gotten the hang of this, try this one on your own. Your DNA sequence is CAT, CGG, TGA. So the first thing, you're going to write your mRNA sequence. So mRNA sequence first, using your base pairing rules, and then your protein, using the chart. Good job, Stephen. Good job, Justin. Good job, Josh. Nick, good job. Dylan, perfect. Jacob, good. Jacob B. Dylan, are you struggling? Let's see here. Dylan, do you understand what to do first? Justin asked, what the first thing you have to do. What is that you wrote? I can't. So remember, you're going to turn that CAT-CGG into an mRNA. So use your base pairing rules first. Skylar, what binds to C? What is it? Okay, so C binds to G. Okay. You have to remember, I think the easiest way is to remember the word at, and then remember it can be or you, depending on whether it's on. Right? Right, but don't forget, you're making mRNA, Skylar, so that's going to be, there's no cheese. So you can even go back in and just replace all the cheese with you. Matthew, do you know what's going on? Okay, so what's the first step? I put it up there, you have to make mRNA. So what do you have to do to make mRNA? Okay, what do we do with the last one? Use the base pairing rules. Skylar was just talking about this. So what binds to C? G. So the first one would be G. What binds to A? T or U, and we're making mRNA, so it's going to be U. Okay? What binds to T? Okay. What binds to C? Good. What binds to G? Good. What binds to G again? And then what binds to T? Good. What binds to G? And what binds to A? Remember, we're making good. U. Hannah, are you putting the sequence in the chat? Hannah, you want to put the sequence in the chat so you get credit for it? So Matt, put that on there, right? Joseph, good job. There's just, I think there's a little, your last one's off, Joseph. All right, so I don't know, Matthew, I don't know where you're getting that from right. So the first thing that you're going to do is make this into an mRNA. So then you're going to separate them into codons. And then you start from the middle. GUA is my first one, GUA, GCC, ACU. All right, so hopefully you got the hang of that. There are practice problems in your book, but I strongly suggest that if you're not, have no idea what you're doing, you go and look at those. And I just want to talk about the last part of the book chapter, which are mutations. So what do you think of when you think of a mutation? Good, bad, indifferent, superhero? What about you guys at home? All right, mutations can be good and bad, right? If you know someone with Down syndrome, that's an example of a mutation. But it's also why we can speak. So the evolution from primates to humans allowed better cognitive features, which are started off as a mutation, as well as the ability to speak, which is, again, a mutation. An opposable thumb is a mutation. So these are all mutations that were just beneficial. So they can be either good or bad or indifferent completely. So there's two types of mutations: gene, which are smaller, they involve only one gene, so they involve one protein, and chromosomal, which are profound. So a chromosomal mutation, sometimes thousands of genes are involved in that. So we're just going to talk about the gene level mutations, and then we'll go into gene and chromosomal mutations in the next section. So gene or molecular level mutations can be two types. You can have a point mutation, which means a single nucleotide changes, or you can have a frame shift mutation, in which the whole reading frame gets kind of put off. So point mutations, which again are the smaller gene mutations, can be three types. You can have a silent mutation, which means exactly what it says. You can see no discernible difference. So when we were doing the translation part, if you look at this chart, you can see that some of them are repeated multiple times. Right? So if I have a mutation in this third nucleotide part, it doesn't actually change the amino acid. So that would be an example of a silent mutation, where you have a change in the DNA, it changes the nucleotide, but it doesn't result in a net impact of the protein. It doesn't actually change the mode of the protein, so it's silent. A missense mutation, yeah.
Did you access example of mutation?
An example of a silent mutation?
Yeah, for an example, like give an example of mutation.
A silent mutation, there's no change. That's why it's silent.
Okay. Did you access for an example of mutation? When we were at mutation, like before this, like before you access, like give me an example of mutation.
No, I was asking what you would think of as a mutation. So, when I say the word mutation, a lot of people think it's superheroes. That's why I was asking, because some people think mutations are very bad, always bad. Some people think mutations are good, but they're both.
What kind of mutation?
No, there's both types. You can have chromosomal mutations that are beneficial, and you can have gene mutations which are beneficial. What's that?
May I go to the back?
Yeah, go ahead. All right, the next one is a missense mutation, which changes an amino acid from one amino acid to another, and results in a change to the overall protein. And then the nonsense changes the codon to a stop codon. So you change an amino acid to a stop codon. So students very often confuse missense and nonsense. Hopefully, maybe one way you can remember it. A nonsense mutation is the word no, so no more protein is being made because the stop codon is reached. Right. So then the other type of point mutation is a frameshift mutation, where you have an insertion, and the book talks about these, or deletion. And what happens with that is it changes the reading frame, right? So we were talking, when we were doing these, imagine if I put an extra nucleotide in there, right? Now I go to make my codons, and it changes the whole reading frame, right? So each one of those amino acids is now different, because it was going to be this. So everything downstream from that insertion or deletion is a different amino acid. And this can be really profound, but it also can create a protein that has some novel characteristics, or allows the organism to do things that others couldn't. Now, it can also happen that you have 3 nucleotides inserted or removed, in which case it's a frameshift mutation, but all it does is result in a different one amino acid being taken out or in, or put in. All right, so the 2 types of mutations are, so Hannah, if you didn't get into the Pear Deck, put it into the chat, otherwise you'll lose credit. Jacob, Justin, Joseph. All right, so most of you got that right. The 2 types of mutations are chromosomal and gene. The 2 types of gene mutations are so gene level. That's point and frame shift. A frame shift mutation is a type of what mutation? Joseph, Justin. That's a gene mutation. A frame shift mutation is a type of gene mutation. It's only involving one gene. When there is a mutation in a gene, but the amino acid sequence remains the same. All right, so most of you got that right. That is a silent mutation because nothing actually happens. When there's a mutation in the genetic sequence that causes a stop codon to replace an amino acid. So a stop codon would mean that there is no longer any protein being made. Okay, so that's a nonsense, no more protein. When there's a mutation in the genetic sequence that causes one amino acid to replace another amino acid. Good, most of you got that right. That's a missense. All right, so you're just going to write a one-line summary about mutations. Don't go into too much detail. What is a little bit you learned about mutations? And if you're at home, please take out your 6.2 review. And everybody here should have a review. And if you need a pen, you can grab it from that container. Remember, please keep it. All right, so we're going to start filling in the review. I put a little up in the upper right-hand corner is what question it is. So this is question 11. We're just going to look at the differences between DNA and RNA. Okay, so DNA has the sugar deoxyribose. It doesn't contain ribose. It's a completely different sugar. The ribose is just the number of carbons it has. They're distinctly different. So RNA contains ribose, and remember that's where it gets its name. That was a test question last section. DNA is always double-stranded. RNA is single-stranded. And for those of you that were struggling to make the mRNA molecules or doing that transcription or translation process, you need to memorize the base pairing rules. A binds to T and C binds to G. So if you were struggling, put a star next to that or outline it or something. Also, remember the T gets replaced when you're making an RNA. So A binds to U. But if you're going from DNA to RNA, the T binds to A. So just, make sure that you understand what the two strands are. So DNA can contain thymine, RNA contains uracil. And most of you should remember this from the last section. We did the same chart then. All right, so this is question 14, as well as question 8. So questions 8 and 14. All right, so mRNA, we already know, is the product of transcription, messenger mRNA, single-stranded, and it's made using the sense strand of DNA as a template. Remember, it contains codons, 3 nucleotide sequences, that correspond to an amino acid. Ribosomal RNA, along with proteins, creates the ribosome. So the ribosome, remember, is really just a scaffolding to hold the mRNA for the tRNA to come in and bring the amino acids. So this was a little animation in your book, if you remember. And then finally, your transfer RNA contains anticodons. So for number 14, number 1 contains an anticodon. It's clover-shaped, and one end holds an amino acid. So this tRNA comes to the ribosome, and the mRNA enters into the ribosome, and you have that codon-anticodon pairing that allows the correct amino acid to be added to the growing protein. And I'll show you a little animation in a second. All right, so this is going to be the first step of translation, which again is called transcription.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome.
Holy slow.
In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA making a strand of messenger RNA out of 3 bases in the nucleus. The DNA code determines the order in which the 3 bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm.
All right, so if you look at question 9, I know that it was a little laggy, but what did we just watch? That's transcription. Put it into your own words. What did you see? What's the point? What happened? What will help you remember transcription?
Mr. Nucci.
Yeah.
Sorry, I heard myself. If you're gonna put move your mic because it's like feeding back or moving.
Okay.
Thank you.
All right, so that's question 9, and questions 12 and 13 we'll go over again on Thursday, but hopefully you're starting to get the process of transcription. So while you're still writing, transcription begins, remember, on the promoter sequence with RNA polymerase unwinding the DNA. So that's important to remember, because there is no helicase for transcription. Then you use the base pairing rules. Remember, C binds to G and A binds to T or U. Everybody, you need to remember that. And there's 3 nucleotide sequences on the mRNA. These are called codons, and each codon corresponds to an amino acid. Sometimes you'll have the introns removed, and we've spoken about introns previously. And unfortunately, a lot of people confuse introns and exons. Introns are what are taken out. They're the garbage. One way to possibly remember it is you throw things in the garbage, introns into the garbage. I mean, because I can't tell you the percentage of people that mix up introns and exons. And then it gets a cap and tail and leaves the nucleus. All right, so this part is translation. I'm going to mute myself. I think it's just bad internet.
A lot more people can hear than we've had in a while.
This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time.
So remember, the tRNA has the anticodon and the mRNA has the codon.
As each triplet is read, a transfer RNA delivers the corresponding amino